welcome. Welcome, everybody. I see there are a fair number of people online as well. We have a huge auditorium today, though. So you can all fit in here if you wanted to come over. So thank you all for coming. I want to welcome everybody. This is our first CART Distinguished Lecture of the year. As you know, we have one once a month on Fridays at 9 a.m. And we have a great series this year, but we're really happy to start it off. And we'll introduce our speaker very, very shortly. I just want to say that it's sponsored by the UCLA Tarjan Center, the IDDRC, the Can Reach program, and the Brain Research Institute. And I want to say that we're recording the talk and we'll post it to the website as well. Let's see, I'm in there. And see what else. Oh yeah, Zoom. Attendees can ask questions through the Zoom, the chat, and we'll try to get to them to the best of our ability. But of course, people who come in person have first dibs. So we're really, really pleased to have Lisa Aziz Zadeh here today. She's a professor, and you can see on her title, the Brain and Creativity Institute at USC. She's the director of the USC Center for Neuroscience of Embodied Cognition. But I'm not going to say any more because Marco Iacoboni, who was her PhD mentor, is going to introduce her. Thank you. And Marco Iacoboni is a professor here in Semel, whom all of you must know, if not through his affiliation with Mirella.
Oh, thank you. There's a great introduction for me. I'm very pleased to introduce Lisa. I've known her almost for my whole life. Well, no, my life is way too long to make that claim. But over dinner last night, we were thinking, so when did we publish the first paper together? It was more than 20 years ago. She was a grad student in psychology here at UCLA, and she did great work as a grad student. And then she went on, did postdoctoral fellowships with Rizzolatti in Parma, with Lakoff, and then back here at the Institute for Creativity, and she was doing work when Damasio came to town and thought, how can I build a great and strong team? Well, let me recruit Lisa. She's been working with Damasio for a while now, several years. The latest thing she's doing really cool stuff funded by Google on rehumanizing the dehumanized. And also this work that she's going to talk today about, which is funded by the DoD, and it's about sensorimotor integration, which is a long time passion. When she was a grad student, she was making connections between sensorimotor integration and language and social cognition, but also applied to, of course, to autism, and also the latest things looking into microbiome and the brain and autism. Okay, I'll shut up here. Lisa, come please.
All right. Thank you so much, Marco. Thank you so much, Dan. It's a pleasure to be here. It always feels like home coming back to UCLA. So thank you. Thank you for inviting me. So today I'm going to talk about two main programs of autism study that we have in my lab. The first one is understanding relationships between sensorimotor and social behavior in autism. And this will be both behavioral and brain data. And then the second thing we'll be talking about the gut microbiome and the brain and how that might relate to autism. Okay, so starting off with the first program. So we all know that about 80% of kids with autism have both sensory and motor issues. And the question that we want to ask is, how are these basic sensory motor functions related to social issues in autism? To answer this question, we're going to compare all of our kids with autism to a control group of kids with dyspraxia or DCD, developmental coordination disorder. So if you're not familiar with DCD, the primary deficit is motor. There are some other comorbidities, but primarily it's a developmental disorder where kids have difficulty making coordinated actions, learning new actions, balance, and so forth. And the question we want to ask is, will we see a relationship between sensory and motor function that's different in autism from DCD? Okay, so for all of the studies that I'll talk about, we have the ends vary depending on the study, but in general, all of the kids with autism are all of our kids are 8 to 18 years old, right-handed. They're all high functioning, so an IQ of 80 and over. And then the DCD kids have no concurrent ASD diagnosis, no first-degree relatives with autism, and we measure DCD by the MABC, which is the gold standard. I'll show you the MABC in a second. And then we also have a third group of TD controls, or typically developing controls. Okay, and the design of these studies is that they come in for a first day of assessments. They also go inside a mock MRI scanner. They get used to the scanning environment. They practice. They learn to hold their head still. And then they come in for a second day where we do functional MRI, structural MRI, and they also bring in a stool sample. Okay, so the first question we can ask is, how do behavioral sensory patterns correlate to sensory behavior, to social behavior in both autism and DCD? Okay, so as you might expect, the ASD group in red, about 74% of the kids have sensory modulation issues, as you would expect. Now, interestingly, also 31% of the DCD kids have sensory modulation issues as compared to 4% of the typically developing kids. So there is a enhanced sensory processing issues in the DCD group, but definitely not to the degree of the ASD group. Okay, and what does this correlate with? As you would expect in all three groups, it correlates with anxiety, aspects of the CBCL. In the ASD and DCD groups, it correlates with repetitive behaviors here. And in the DCD group only, it correlates with motor ability here. So that's the green. Okay, so what about social? Interestingly, only the ASD group shows correlations with social ability. So with alexithymia, here in red with the IRI, perspective taking, which is a measure of cognitive empathy. And then here with social ability as measured by the SRS. Okay, so only in the ASD group are these basic social modulation difficulties associated with social functioning. Okay, can we find a similar pattern with motor behavior? Okay, so first of all, we ran the MABC, which is the gold standard for dyspraxia for DCD. And this is things like manual dexterity, turning pegs, aiming and catching, which is ball throwing skills, and then balance. And when we look at the two clinical groups, both of them have difficulties compared to the TD group, but they don't differ from each other. You can't tell them apart. So most of our kids with autism show very similar manifestations of motor ability compared to the DCD group on this measure. When we first received the R01, Jonathan Delafield's butt in the UK had published this study showing that if you have kids play this very simple coloring game on the iPad, the kinematics from their movements is recorded by the touchscreen on the iPad. And using the touchscreen sensors and the IMU sensors of the iPad to measure the kinematics, you can run machine learning on that, and you can distinguish the TD group from the ASD group with about 93% accuracy. So we saw that paper and we asked him if we could use his game to see if we can distinguish the DCD kids from the ASD kids. So we did that, and we found that you can use these basic kinematics from just this coloring game and use machine learning on it to distinguish the two clinical groups, ASD and DCD, with about 70% accuracy. And we found that those differences in kinematics actually correlate with cerebellar activity in the CRUISE-1 and CRUISE-2. Okay, so there's something about the way the kids with autism are moving that is different from the kids with DCD. The other motor test that we ran is the Florida Apraxia Battery. So here they're asked to do 3 different things. One is gesture to command, so show me how you use a blowhorn. Imitation, which is 2 forms of imitation, meaningless imitation, which is something like copy what I do, and they put their hand to their ear, or copy what I do, and they comb their hair. So that's meaningful imitation. And then the third one is tool use. So you actually give them a pair of scissors and you say, show me how you would use this. And what we find is that there's only one of these measures that distinguishes ASD from DCD, and that's imitation of meaningful gestures. So only when you do something like copy what I do, when you're doing like combing your hair, do they show spatial and timing errors, the kids with autism, significantly more than the other two groups. And then the really interesting part is that the number of errors that they have correlates with affect recognition. So how well they can tell other people's facial expressions is correlated with their meaningful imitation ability. Okay, so just to summarize these behavioral studies, only in autism do these sensory and motor differences relate to socio-emotional differences. We don't see that in the DCD group. With sensory, that was with alexithymia, social skills, perspective taking, and with imitation, affect recognition. Okay, so now we want to move on to the brain and see if we can understand what's going on in the brain that leads to these behavioral differences. We know that there's a ton of studies in autism versus TDs. In general, they find a lot of different things, but some of them are like the inferior frontal gyrus, the cerebellum, the corpus callosum. But we all know that those are also motor regions. So is what they're picking up actually related to the motor difficulties, the comorbid DCD in autism, or is it actually related to autism itself? And so we have a DCD control group, and by having the DCD control group, we can try to tease apart what's actually comorbid DCD and what's actually autism. Okay, so here's the tasks that we had them do inside the fMRI. So they're watching these videos of emotional facial expressions, non-emotional facial expressions, and hand actions. And they have 3 different tasks. So in the first run, they're asked to watch the video. So all they have to do is watch the videos. This is called action observation. They imitate the videos, imitate what you see the actor do. That's the imitation run. And then the third run is think about why the person is making the action. So this is mentalizing. Okay, and I'll show you a video of our stimuli. And all the hand actions are bimanual. Okay, so either they just watch it, action observation, they copy it, they imitate it, or they think about why the person is doing it. Okay, so first of all, when they're doing the imitation task, we get autism less than TD and then inferior frontal gyrus, which is consistent with previous data. But interestingly, we get the same exact thing in the DCD group. So we think that this hypoactivity in the IFG, as well as the SMA and premotor cortex, is really related to the comorbid motor issues that they have. Okay, so here in white is the TD group. Here you can see the ASD group has hypoactivity in the IFG, but also so does the DCD group. Now, interestingly, what we find is the difference is action observation. So when they're asked not to do anything, just watch the videos, here you see the ASD group has hypoactivity in the IFG compared to the typical group, but also compared to the DCD group. So the DCD group shows no differences compared to the typical group, only the autism group shows hypoactivity compared to both groups. Okay, what is this related to? We find motor skills relate to activity in the IFG for all tasks. And this is consistent with previous data showing that activity in this region is related to motor expertise. So the more expertise you have, the more activity you have in this region. And then interestingly, we also find that activity in this region is correlated to autism severity as measured by the ADOS and the ADIR. And this is even after controlling for motor ability. Okay, so we find that in autism, there's this unique hypoactivity in the IFG only for action observation. And then the question is, why does that happen, right? So what's the connectivity differences that is leading to this hypoactivity? Here's the IFG, and here's the visual cortex. And we know that there's two pathways from the visual cortex to the inferior frontal gyrus. There's this dorsal pathway through the superior longitudinal fasciculus, or the SLF3. And then there's this ventral pathway through the extreme capsule and the inferior frontal occipital fasciculus, or the IFOF. The dorsal pathway is more of a motor pathway, and the ventral pathway is more of an emotional conceptual pathway. And so what we hypothesized is that maybe both the ASD and DCD groups, since they both have motor difficulties, have some less diffusivity or white matter in differences in the dorsal pathway, but only the autism group would have less diffusivity, less white matter connectivity in the ventral pathway. Okay, so first of all, when we just look at autism versus typicals, we find the cerebellum, the cingulum, and the interesting thing is that also the DCD group shows the same thing. So these seem to be regions that are related to motor dysfunction rather than just to autism. However, we do find these unique patterns in autism compared to both clinical groups. And this is really in the cingulum, both the longitudinal fibers of the cingulum and the U fibers. I'll show you this in a second. And then we find these other patterns where the autism group is significantly different than both groups as an extreme group. And this is in the middle cerebellar peduncle, the frontal part of the corpus callosum called the forceps minor, anterior commissure, and then also the posterior IFOF in autism compared to TD. Okay, so let me just show you what this looks like. So here are the unique differences in autism. Here are the longitudinal fibers of the cingulum as well as the U fibers. This middle frontal part of the cingulum. Here is the anterior part of the corpus callosum, the forceps minor, and here's the middle cerebellar peduncle. So all of this is more different in autism compared to the other two groups. And then here is the IFOF, the posterior part of the IFOF, which shows more diffusivity differences in autism compared to DCD and TD. Okay, so what does this relate to? Differences in the frontoparietal cingulum is related to autism severity, as well as to alexithymia. Differences in the middle cerebellar peduncle are related to autism severity and autism severity, and in the IFOF with alexithymia and autism severity. Okay, so this is structural connectivity. What about functional connectivity? We know from previous work that this IFG region is related to a lot of other regions when you're asked to think about why the person is making the action. So when you're doing a mentalizing task, there's a lot of crosstalk between this region and theory of mind regions in blue here. And this is a study by Spunt and Lieberman where they show that when the participant was asked to think about why someone was making an action, the inferior frontal gyrus was functionally connected to mentalizing regions like the medial prefrontal cortex, the TPJ, the precuneus, and so forth. Okay, so now we wanted to see if we'll also see these functional connectivity differences when we compare autism to the two other clinical groups. Okay, so here again is our task. So they're looking at these videos and they're asked to think about why the person is making the action. And we find that when we use the IFG as a seed, we show less functional connectivity with the left TPJ, a mentalizing region, compared to both typicals and DCD, and more connectivity with emotion regions like the insula and executive functioning regions like the DLPFC. Okay, so more functional connectivity with emotion and DLPFC, and less connectivity with mentalizing regions like the left TPJ. Okay, so to summarize this set of studies, even though kids with autism have 80% of them have motor difficulties, they have DCD, autism has a unique motor profile. And this is related to timing and spatial errors with meaningful imitation, which is related to social skills like affect recognition. And you can use a simple iPad game to tell apart the kinematics from the kids with autism from the kids with DCD. We find the inferior frontal gyrus is hypoactive only in the ASD group during action observation, and that's related to autism severity. We find when mentalizing, the inferior frontal gyrus has less connectivity with the mentalizing network and increased connectivity with the insula and DLPFC. And then we find that compared to the other two clinical groups, autism has unique diffusivity differences in the cingulum, frontal corpus callosum, middle cerebellar peduncle, and IFOP. And these relate to emotion processing and autism severity. Okay, so for the next part of my talk, I want to focus on the gut-brain microbiome system in autism. And for this part of the talk, we'll only talk about ASD and TD. So we're going to drop the DCD group. Okay, and so we're going to talk about brain behavior, and then we're going to talk really about metabolites from the gut microbiome, and specifically from the tryptophan pathway. Okay, so just to make sure we're all on the same page, the gut microbiome system is a link between the brain and the enteric nervous system. So the enteric nervous system is a network of neurons that lines the digestive system, the digestive tract. There's 200 to 600 million neurons. So people talk about this as evolutionary being our first brain. Just to give you a sense of scope, the spinal cord has about 100 million. So this is 2 to 6 times more than the spinal cord. Okay, now these neurons, most of them, 90% of them are actually afferents. They're going from the gut to the brain. And what they're talking to in the gut is the gut microbiome and metabolites from it. In fact, most of our neurotransmitters are metabolites from the gut microbiome. Okay, so how does this relate to autism? There's now many studies showing dysbiosis of the gut microbiome and how that correlates with autism severity. There's a lot of rodent studies that show if you give a fecal transplant to mice from children with autism, they develop autism-like behaviors like repetitive actions, antisocial behavior. And then interestingly, when they're given a second fecal transplant from a healthy donor, their symptoms are reduced. And then there's a small sample study showing that kids who were given with autism who were given vancomycin, which is an antibiotic, had short-term improvements in autism. And also fecal transplants seem to improve symptoms in a small sample of children. So these are very preliminary studies, right? A lot of them are small samples or mice studies, but there seems to be some relationship that might be actually causal in autism. Okay, so by now there's so many studies looking at the brain with symptoms in autism. I shared some of those today. And then there's also a lot of these studies coming out now showing relationships between the gut microbiome and autism symptoms. But what we really want to do in our program of study is to do this, to look at how the gut microbiome interacts with the brain, right? Modulates brain activity, and then that leads to symptomology. Okay, and so our hypothesis is that there's some kind of altered communication between the gut microbiome and the brain. And this modulates brain activity, which via altered metabolites and specifically tryptophan pathways. And then this modulates behavior. We focused on the tryptophan pathway because there's long been the serotonin hypothesis in autism. We know about 30% of kids with autism have hyperserotonin levels. This is associated with social and communication issues, as well as GI issues, which a lot of kids with autism have. Okay, so here's your dietary tryptophan. In the gut, this becomes serotonin here. This is the serotonin pathway. This is via microbiota microbiome host interactions. Another part of this pathway is chiurenine. This actually has, in different parts of development, neurotoxic effects. It's correlated with anxiety and depression. Chiurenic acid and picolinic acid actually have neuroprotective effects, and these tend to be lower in kids with autism compared to typicals. And then there's also the indole pathway, which many people now have found differences in abundance in autism and have related to that to autism severity. Okay, so looking at our groups, just behaviorally, we find that there are differences in sex, FSIQ, BMI, and so we use all of these plus age as covariates in all our analyses. We find differences in prenatal antibiotic usage, so ASD more than TD. The kids with autism tend to have more GI issues. And then all of this is what you would expect, but I want to focus here on the differences in discussed propensity and sensitivity are actually greater in autism compared to typicals. And disgust was something we wanted to focus on because we thought of all the behaviors that might be related to the gut, disgust is probably one of the strongest, right? So we'll talk a little bit more about disgust. Okay, so just looking at metabolite differences between groups, and I just want to say also all of our groups, they had no pre or probiotics for 2 weeks before took the study and no antibiotics. Okay, so just looking at metabolite differences, we find significant differences in chiurenate between the two groups. So it's less in the ASD group, and this is a tryptophan metabolite. When we run machine learning on the metabolites to try to discriminate the two groups, there's 2 metabolites that can significantly discriminate them, and those are chiurenate, which is a tryptophan metabolite, and arginine, which is a glutamate. If you just run it again with just the chiurenate, it also comes out significant. Yes, it's all from stool, yes. No, that's a great question. Okay, so now looking at relationships between metabolites in the brain, we focus, in this talk, I'll just focus on 2 of the tasks. So looking at emotional faces and non-emotional faces, because we know this is a task that shows reliable differences in autism. And also the disgust processing task. So again, we wanted to look at tasks that related to vagal processing, like disgust, and we know show differences in autism. So either they looked at disgust facial expressions or disgusting foods. And this is all fMRI. Okay, so the first task, when they watch these videos of facial expressions, we find hypoactivity in the ASD group in 2 regions, the inferior frontal gyrus, which we talked about a lot previously, and also the middle cingulate here. And we find that activity in these 2 regions is related to autism severity and disgust propensity. And we find that activity in the inferior frontal gyrus is related to anthranilate, which is a tryptophan metabolite, and in the MCC with tryptophan betaine and C-glossitrophan, tryptophan, sorry. Okay, so there's a relationship with how much of these metabolites you have and how much activity you see in these regions in autism. Okay, looking at the disgust tasks. So here, again, they're looking at disgust facial expressions or disgusting foods. We see differences in autism versus TD in the mid-insula and the dorsal anterior insula in this task. These relate with autism severity and sensory sensitivities. And then they also, the activity in these regions correlates with indolactate and chiurenate. So here, the right middle insula with indolactate and the left dorsal anterior insula with chiurenate. Okay, and so now we want to test our mediation models. And what we find is that here's the mid-insula region. It correlates with indolactate. So here's the right middle insula activity. It correlates with indolactate. And it also correlates with autism severity as measured by RRBs and the ADOS total. And we find the mediation model is significant for both of those. Here is the mid-cingulate. It correlates with tryptophan betaine and with discussed sensitivity. And then we find that the mediation model is significant also here. So tryptophan betaine is modulating MCC activity, which in turn is modulating discussed sensitivity. Okay, so to summarize this set of data, tryptophan metabolites correlate with known autism functional brain differences. So these are regions not just that we've found, but many, many people now have found are different in autism. And the activity in them correlates with these tryptophan metabolites. The mediation model is significant. So it seems like the abundance of these metabolites is affecting or modulating brain activity, which in turn is modulating behavior and symptomology. We still have a lot to do on this. So we're now currently running shotgun metagenomics, just to give you a taste of that. So we're actually looking at the bacteria. That's what shotgun metagenomics does. So not at the metabolites, but the actual bacteria. And we see differences in TD versus ASD. And one of the bacteria that shows a difference is Escherichia, which is related to tryptophan metabolism. We also collected saliva samples from all of our participants. So now we can look at the oral microbiome, relate that to the gut microbiome, relate that to the brain and to behavior. So look at a fuller picture of the system. And we can also do all of this with our DWI and resting state data as well. So we still have a lot to do, but the first steps look really promising. Okay, and that's about it. So I would just want to acknowledge all the people that helped us, my lab, all of our participants, our wonderful collaborators at UCLA, Morella, Susan, Emron, Jen Leibus, Annie Gupna, at CHLA, and at the University of Parma, who helped us with our DWI data. All right, and I'm happy to take questions. Thank you.